Hi, I'm Lauren Sansing, and I'm here with Ray Koehler, Francesca Bosetti, and Cenk Ayata, and we're here to talk about a little bit more of a deep dive into the SPAN network. Francesca, is it okay if I start with you and ask you, how did the idea begin?
Yeah, so the idea began with the NINDS effort to address three important issues for NINDS and for the scientific community. The first one is that most studies in preclinical stroke research and other fields, for that matter, mostly used young animals, small sample size. They were not done in a randomized and blinded fashion. So the predictive value of these studies was perhaps not as strong as we desired. So here came the idea of a preclinical stroke network that could do experiments within a highly rigorous fashion, with including randomization, blinding, and using male, females, using animals with comorbidities, trying to mimic the population of patients that have ischemic stroke, and also trying to mimic the methodology that is used in clinical trials with a coordinating center taking care of randomization, blinding, and drug distribution and statistical analysis. The other important need was to try to prioritize interventions that were proposed for clinical trials. How can we determine if one intervention is more promising than another? And so a SPAN became a tool to try to prioritize these interventions, as we have the ability of comparing directly the efficacy or non-efficacy of different interventions that are tested at the same time. And also, I have to briefly acknowledge that there is an international interest in this space, and a European group named Multipart really started to think about this problem and did a planning effort to address multi-center preclinical trials for stroke.
Hey. Just a quick follow-up question. You designed it as a cooperative agreement, right, with really heavy partnership with NINDS and what was to become the SPAN investigators. Can you talk a little bit about NINDS's involvement in SPAN?
Yes, NINDS really wanted to be involved and make sure that the goals and the procedures of the network were aligned with our internal goals. And again, this is more similar to the way that clinical trials through StrokeNet are administered, and so it becomes a partnership between NIH and the community and the grantees to make this a success. And I must say that SPAN 1.0 worked very well, and really all the goals were achieved. And as you may have heard from yesterday at the main session, we had an intervention that was above the efficacy boundary. And so based on the success of SPAN 1.0, we have decided to invest in SPAN 2.0 and continue this effort with, again, 6 academic sites and 5 interventions that will be announced in the next few months to be tested again by the network.
So Ray, let me ask you, as one of the PIs that were on the ground when we first got started, what were some of the key decisions, and how do we approach those decisions?
Right. So this was a real challenge because we had 6 different sites with 6 different PIs who are used to just running their own biodiscovery lab where they do a stroke in an animal in a certain way. And we had to figure out how to harmonize all of the protocols. Even though most of us do the stroke in animals sort of the same way. There are always slight variations, and so we had to work that out. We also were given the task of having an endpoint that was based on long-term behavior rather than the typical short-term infarct volume. And so it took quite a while to come up with a decision of what behavior test would be best, because we know that, in clinical trials, it's the modified Rankin at 90 days. So we just didn't want an acute outcome. So we had chosen a test called the corner test because it does not show compensation over a month of time. And then, and we were doing this, and then the pandemic came and we were shut down for 6 months. And so we didn't have enough time to pilot everything. And we just had to jump in October of 2021 and do the study in 21 months. And so we were really on a treadmill at that point. So it was exciting. And then the other task we were given was to figure out what models of comorbidity we should use. And even though there were pieces in the literature of different models, we didn't know which one would be easiest to implement at 6 sites for high throughput, because we were going to be doing, approximately 600 to 800 animals per comorbidity for a total of 2,600 animals. So that took a lot of give and go to figure out what would be feasible to do at 6 different sites. So it was interesting experience, let's say, to organize all of that.
And it's nice that we can leverage that experience moving into SPAN 2.0.
Exactly.
So at least we have a running start as we move forward. Cenk, do you want to talk a little bit about how we decided to measure tissue injury?
Right, yeah. So even though our primary endpoint was a behavior endpoint, just like in any experimental study or clinical trials, in fact, some clinical trials, we had to have some handle on the tissue outcome as well. So that wasn't an easy decision because there are basically 2 ways to go. One could do the traditional looking directly at the tissue using histology, tissue sections, or typical histochemical stains. The problem with that, though, is twofold. One, you can only do it once in one animal. Once you take out the brain and look at it, you're done. So you couldn't do longitudinal studies. The second problem is we don't do that in our patients. Our patients' tissue outcome is assessed using MRI. So putting that together, we looked into the possibility of doing MRIs in all animals and decided to do that at 2 different time points. First, at 2 days, which is an early time point where infarct maturation is generally accepted to be complete so that we have an infarct volume handle. And then at 30 days to look at the tissue volume to quantify the atrophy. There are several ways of doing that as well. The downside of having MRI was the logistics, of course. Every site had to have an MRI, small animal magnet, and they had to have expertise in looking at stroke images. And we had to do a very large number of MRIs. We planned to do over 2,000 animals. That would be over 4,000 MRIs, and the cost became a limiting factor. But, we did the cost-benefit analysis, looking at the manpower needed to do the histology, doubling up the animal numbers. We wanted to do late time point in animals because the behavior had to be at late time point. But infarct could not be assessed at late time point using histology. So altogether, we decided to go with the MRI. Number 1 factor, in my opinion, was the clinical translatability.
Yeah.
Number 2 factor was longitudinal imaging possibility, cutting the animal use by half.
Francesca, maybe I'll just end with a question to you, which is, there were some changes made for SPAN 2.0. Can you talk a little bit about what those changes are and what you hope from the next phase?
Yeah, so the first change that I think is important to highlight is the terminology that we used. We are used to the term neuroprotection, and for SPAN 2.0, we moved away from the word neuroprotection and chose instead cerebral protection, or even better, cerebrovascular protection, because we are looking at interventions that go beyond the protection of neurons, but they protect the brain cells and the vasculature in their entirety. The second change was the separation of interventions and the testing laboratories. In span point 1, the applications from the sites had also to include an interventions, and we thought that was perhaps limiting the promising interventions that could come in, or qualified labs that had the capability of being a very good site. So by separating them, we really hope to increase the quality and the volume of the applications so that we could have quality applications for interventions and quality applications for testing labs. And finally, the third new aspect is that we opened an RFA specific to small businesses to again encourage this partnership between NIH, the scientific community, and the small businesses, and give again the opportunity for promising interventions to be tested in this rigorous fashion by the network. And if everything goes well with the awards, we'll have some small businesses participating in SPAN 2.0.
I look forward to it. Thanks so much for it, guys. It is a new era, and I think it was SPAN 1.0 was a great success. It was a lot of work, but I think we learned a lot, and hopefully. Well, and now we have a huge data set to share with the community and do additional analyses. And I look forward to moving forward. All right. Thanks for being here today and sharing your thoughts.